Okay, well, as I think most of you know, your exam is ongoing. I've had a couple emails from people who, in disbelief, are not sure that the exam is actually closing Monday. They want to be sure. Yes, it's true. You can have till Monday to take the exam. I have office hours today and again on Monday. All right, so, but we're going to move ahead today. We've spent the last few chapters talking about the major classes of biomolecules, so proteins, amino acids, carbohydrates, lipids, and nucleic acids. And chapter 27, which I think we're going to spend 2 class periods on, maybe just a little bit more, is on metabolism, so how nature uses those molecules and does reactions with them to generate things you can use to drive other processes. The currency that we're going to be sort of using as we do reactions with these biomolecules are first ATP, which you know of. This is the major energy carrying molecule in biology. You've got 3 phosphate groups linked to each other via what are called phosphoanhydride bonds. And you've got these 3 phosphate groups of ATP, which are called alpha, beta, and gamma. And a phosphoanhydride linkage links beta and gamma and alpha and beta. Those are fairly unstable bonds, and in the same way that an anhydride is fairly unstable. Then for ATP, the alpha phosphate is linked to the 5' hydroxyl of ribose, and then you have the adenine base here, which we'll just abbreviate. The other molecules that we'll be dealing with, and we'll draw their structures a little bit later in terms of currency that we'll use in metabolic reactions, are NADH and FADH2. These are reducing agents, but they also are electron carriers, and so they can carry electrons to the electron transport chain in mitochondria. We're not going to talk about that at all in this class. You'll have to learn about that in biochemistry. But in the electron transport chain in mitochondria, these molecules drive a proton gradient across the mitochondria, which comes back through an ATP synthase and generates even more molecules of ATP, which of course your body can use for basically for all processes that require energy, for motion, for reactions, and so on. So we'll see that all 3 of we won't talk about nucleic acid metabolism, but we're going to see that lipids, carbohydrates, and also proteins can each be funneled through metabolic processes that can result in ATP and NADH and FADH2. For example, if we're just focusing on the triglycerides, where you have a glycerol molecule and then 3 fatty acids, as we've talked about. The process involves an enzyme called lipase, which hydrolyzes the esters to liberate the 3-carbon alcohol, which, as I said, is called glycerol. And then the 3 fatty acids. And these undergo a process called fatty acid beta oxidation, which we're going to talk about in detail in just a few minutes, ultimately clipping off 2 carbon units, several 2-carbon units from these fatty acids to give you this 2-carbon thioester called acetyl-CoA. Glycerol ends up getting modified in a way that we'll talk about and interfaces with carbohydrate metabolism. So for carbohydrates, here's glucose, and we'll talk about how glucose beta-D-glucopyranose undergoes a process called glycolysis, which converts the 6 carbons of glucose into 2 3-carbon molecules here called pyruvate. And it turns out that glycerol can get modified through a couple of processes that we'll talk about, and can enter into glycolysis at an earlier stage. So you've got 6 carbons in glucose, you've got 3 carbons in pyruvate. So for every glucose molecule, you get 2 of pyruvate. And then pyruvate undergoes something called oxidative decarboxylation. We're going to lose CO2, 2 molecules of CO2 for every molecule of glucose that goes in. And we'll again end up at this juncture where we have acetyl-CoA. So you can see that nature's efficient. We're funneling different starting materials to the same junction point, and then we're going to do some more specific things with acetyl-CoA. Proteins, of course, are just polymers of amino acids, and we won't say too much about amino acid metabolism, though in the problems that you've had so far with PLP and dealing with imines and amino groups, that's part of the story. We won't talk about that in a lot more detail than we already have. Proteases clip the amino acids into, the proteins into their component amino acids. And then for each amino acid, there's different, to some extent, different reactions that are used, but you've got basically amino acid metabolism that's going to interface with either acetyl-CoA or with some other things where you can funnel amino acids through this pathway. We're not going to talk about amino acid metabolism a lot. In fact, we'll just leave that for biochemistry and future classes. Once you get to acetyl-CoA, there's a process called the citric acid cycle, or the tricarboxylic acid cycle, or the Krebs cycle. And you'll see why this is a cycle later on when we describe it, but at various points we're going to lose CO2, we're going to make ATP, or actually it ends up being GTP, which is basically an equivalent molecule to ATP. And also we're going to make NADH and FADH2, which, as I said, we can take to the electron transport chain and ultimately cash in for more ATP. All right? So all of this is going on all the time, and when you hear people talk about burning calories, this is probably what they mean, is all of these metabolic processes leading to molecules that can drive other processes. So that's our broad overview of metabolism. As I said, we're going to start today with lipid metabolism, then we'll move on to carbohydrates. We will talk about the citric acid cycle. We're not going to deal with amino acid metabolism very much. And this will probably take us 2 class periods to get through. Just as a warning or strategy, you can, in this section, power through by sheer memorization alone. I don't recommend that. Memorizing is useful, but only the things you have to memorize. If you can try to focus on how the reactions we're going to look at are connected to trends and reactivity that you've already seen, that's going to be most effective. All right, so let's start out with lipids and metabolism of triglycerides. And let's start with this reaction of a triglyceride to hydrolyze. Oh, that's going to be fun. Are they really going to do that the whole time we're here? Anyway, let's talk about hydrolysis of those esters to get the alcohol and the carboxylic acid. So I'm going to introduce something that you may have seen before. Your text has talked about it maybe once or twice before. We've sort of skipped over it. You'll get it again in biochemistry. And so I don't want to spend too much time on it, but this is going to be a close-up view of an enzyme active site and what the amino acids in the active site are actually doing. And for now, I'm going to abbreviate this as simply an ester, even though you're going to realize there's 3 esters in the same molecule. For the purpose of drawing out the mechanism, we're not going to draw more than we have to. So we're going to do something that I sort of hate, which is represent the enzyme lipase as a blob called lipase. The structure of the amino acid sequence is known. For a lot of enzymes, the 3-dimensional structure is also known. I'm going to just simply focus in on the active site, which has an aspartic acid residue, which in the active site exists in its negatively charged conjugate base form. Then there's a histidine residue. I'm writing down the amino acid number in the sequence, so you can see that these amino acids are actually very far apart in primary sequence, but the tertiary structure of the enzyme brings these amino acids close together in the active site. So a big portion of what enzymes do is pre-organizing all the necessary functional groups in the right place. The last amino acid is serine 132. I do this, as I said, writing down these numbers only to illustrate how far apart in primary sequence these individual amino acids are. Someone's going to ask, do I need to memorize these amino acid numbers? No. Only if you want an A in the class, but otherwise no. No, you don't. I mean, that's a silly question. I'd rather ask you harder questions than that. Memorizing numbers is easy. Okay, so you have these 3 amino acids, and then you have your ester. The ester is placed at a location that has some hydrogen bonding groups adjacent or near the carbonyl oxygen. These are, I think, backbone amide bonds. They basically act to prepare the carboxylic acid to be attacked and to stabilize the resulting tetrahedral intermediate. This group of 3 amino acids in the active site is called the catalytic triad, and its purpose is proton transfer. And so you're going to see some people refer to this as proton shuttling. You're going to see that the aspartic acid removes a proton from histidine so that histidine can remove a proton from serine, so that serine becomes nucleophilic enough to attack the carbonyl carbon to give us a tetrahedral intermediate. All right? So why do those 3 things need to happen at once? Well, think about the pKa's. Serine has a pKa of 16. Histidine, acting as a base, we're thinking about its conjugate acid, which has a pKa of 6. Normally, you would not think that histidine could remove a proton from serine. What pays for that is the fact that aspartic acid is removing a proton from histidine. The other thing that pays for that is the fact that we're inside an enzyme where pKa's change and are a little bit different than they are when things are exposed to water. Also, the strength of hydrogen bonding between these amino acids drives the process forward. So, and you'll see this kind of thing again and again in biochemistry. The mechanism I'm showing you with this catalytic triad, there's actually tons of enzymes that work this way. A lot of proteases do as well. So after that first step, this hydrogen is now going to be on the aspartic acid. And this hydrogen, which used to be on serine, is now going to be on histidine. So we sort of just traded who has the protons. Here's your tetrahedral intermediate. Remember, we're trying to hydrolyze an ester. And the next step of ester hydrolysis is leaving group leaving. The glycerol portion of our molecule, I'm going to highlight in yellow. That's going to be our leaving group. And we're now going to see how the catalytic triad helps the leaving group leave. Electrons on the negatively charged oxygen, which, as I think I pointed out, is being stabilized by hydrogen bonding with nearby groups. Electrons are going to kick down and the leaving group leaves. As it's leaving, it's going to pick up a proton from the histidine, which is itself going to pick up a proton from the aspartic acid. And that's going to sort of reset the catalytic triad. And it also releases glycerol. So now afterwards you have one alcohol of glycerol released. Of course, for a given molecule of glycerol, you've got 3 different hydrolysis reactions to do. Fortunately, the enzyme's really fast, and we're only going to take you through one of those hydrolysis reactions. So if you are following along, it looks like as far as the histidine and the aspartic acid are concerned, we're back to where we were at the beginning. Only now, the carboxylic acid portion of our fatty acid is no longer attached to glycerol, but it's enzyme bound. Okay, we've now traded one ester for another. If we stopped there, the enzyme would be dead. So we now have to hydrolyze this new ester off the enzyme. And nature's going to use water to do this. Any questions so far about what we're doing? Okay. So look at what we did in the beginning. We used aspartic acid to deprotonate histidine. We used histidine to deprotonate serine. We used serine to attack. Now we've got the fatty acid on serine, but we're going to now use aspartic acid and histidine to deprotonate water so water can attack that ester. So again, lone pairs on the aspartic acid deprotonate histidine. Histidine deprotonates water, and water attacks. Again, I'm not going to draw the residues that are hydrogen bonding with the negatively charged oxygen. We'll just continue on. So here's again the pink proton, and we'll make the proton on water blue. After that reaction, the pink proton is on the aspartic acid. So in particular, it's that pink proton that just gets traded back and forth between the aspartic acid and the histidine. Here's the blue proton from water. And then here is the tetrahedral intermediate that you get when the OH group from deprotonated water attacks the serine-bound ester. Then, as we've done before, leaving group, we're going to kick off the serine, negatively charged electrons kick back down. We're going to kick off the serine, and as it leaves, it's going to pick up the proton from histidine, which in turn is going to pick up the proton from aspartic acid. That's going to leave everything in lipase as it was at the very beginning. So if you go back and compare at the start, aspartic acid was in its negatively charged conjugate base form. That's true here. Histidine 263 was in its neutral form. Oops, shown here. And then serine was also in its neutral form, shown there. And the blue proton on serine, the pink proton on histidine. And then here is our liberated carboxylic acid. That's the fatty acid portion of our triacyl glycerol. Okay, so that's the catalytic triad. And if you follow the pink and the blue, you can see we're basically just trading who's got protons and using that to drive the process. All right, questions so far?
Does it only bind to one, the lipase is only bind to one part of the triglycerol?
Yeah, you've only got one catalytic triad, so you can only hydrolyze one ester at a time. And I'm not sure whether that triglyceride stays in the active site and just moves down one until you get all three, or whether it's constantly associating and dissociating. That's probably known, I just don't know it. Okay, others? All right, let's talk about the fate of glycerol. This is this 3-carbon alcohol. We'll talk about what happens to the fatty acid, but before we do that, let's just deal with the 3-carbon alcohol. Sorry, I'm getting mixed up on how to draw it. Our goal is ultimately to convert glycerol into dihydroxyacetone phosphate, which is the following molecule, dihydroxyacetone phosphate. This is one of the intermediates in glycolysis. And so you can sort of tell what we have to do. Here's your 3 carbons, there's your 3 carbons. We need to oxidize this OH group from a secondary alcohol to a ketone. And we need to convert this OH group into a phosphoester. So the thing that happens first is making the phosphoester. So the enzyme that catalyzes this is called glycerol kinase. As you get into biochemistry, you're going to start recognizing what enzymes do by their names. The name kinase refers to an enzyme that transfers ATP to, in general, an OH group or some other nucleophile. Kinases generally make phosphoesters or transfer a phosphate group from ATP to something else. So I'm going to use a curvy arrow to indicate that ATP goes in, a phosphate group is transferred, and ADP comes out. And the molecule that's made at this point puts a phosphate group on one of the OHs. So this bond that we just made is a phosphoester. I've shown the phosphate group in this protonation state. The pKa of this proton here is about 7, so at physiological pH it's about a 50-50 mixture of acid form and conjugate base form. It's not really important which way you draw it. This is called glycerol 3-phosphate. And this step is downhill in energy, but it represents an investment of 1 molecule of ATP. Now, looking at that, this seems like a step backwards, because ultimately in metabolism, we want to get molecules of ATP out that we can use for other purposes. And so consuming ATP to do a reaction seems like a step backwards. And this reflects an important portion of what you'll see in glycolysis. There, in the early stages of glycolysis, we're actually going to invest a few molecules of ATP to get the process going. The payoff's going to be greater than our investment, so we make a profit, which is good, because otherwise we wouldn't all be alive. But you'll see that there are times where you do have to invest ATP. All right, let's talk about mechanism for this kinase reaction. What I'm going to show you is going to be basically the same mechanism that we're going to use all over the place for similar types of kinase reactions. We're going to need a base to remove a proton from glycerol. And then we're going to need to draw ATP again, so we'll just do that real quickly. Here's the 3 phosphate groups. Here's the ribose ring. And then we'll draw the heterocyclic purine base for ATP. And it can be good to just think about the differences between the alpha, the beta, and the gamma phosphates. Most of these enzymes that use ATP are magnesium dependent, and magnesium often is associated with at least some of the negative charges of the triphosphate group in ATP. It sort of helps things be a good leaving group. Which ones sort of doesn't matter, and it may depend on which phosphate group is getting transferred. The reaction that we're showing you here is going to involve a base removing a proton from glycerol. As that oxygen-hydrogen bond is broken, the resulting negatively charged oxygen is going to attack the gamma phosphate group. This kind of thing where a proton is removed at the same time as attack happens is really common in biochemistry, is really common in enzyme catalyzed reactions. It's not always that way. Sometimes you have to remove a proton first. Sometimes you have to remove a proton after attack. We're not going to be really careful about that. Nor are we going to be careful about what the group is that removes the proton. It could be water, it could be an enzyme active site. You've seen at least some examples of what that can be up here with the lipase type chemistry. But the the oxygen on carbon 3 of glycerol acts as a nucleophile to attack the gamma phosphate group. What happens or what is shown next will vary depending on the source that you're looking at, depending on whether people are going for accuracy or just a quick summary. There's evidence that the chemistry at the phosphorus is sort of like carbonyl chemistry. In other words, we're going to show making a new bond between oxygen and phosphorus at the same time as we show a phosphorus-oxygen pi bond breaking. This is kind of obnoxious because it requires us to do another step, Some people, instead of showing this addition, elimination chemistry like I'm going to show you, will just show this as though it's an SN2 reaction. So some textbooks are just going to show and the leaving group leaves. But I'm trying to be consistent with what's shown in the Smith text and what the evidence suggests. So we're going to do it that way, even if it's a little bit more challenging. So nucleophile attacks phosphorus. We're going to get not a tetrahedral intermediate, but an intermediate where that gamma phosphorus is attached to 5 different things. That's okay for phosphorus because it's further down in the periodic table and it's larger and can form strong bonds with more than just 4 things. All right, so this is instead of a tetrahedral intermediate, we might call it a pentavalent intermediate. This is the gamma phosphorus, and then here's the beta phosphate group and the alpha phosphate group of ATP and the 5' hydroxyl of ribose, and then we'll just abbreviate the base A. Here's your magnesium that's chelating or binding to the negatively charged oxygens on the beta and alpha phosphate groups. Then that tetrahedral intermediate is going to collapse. Everything here is going to be a leaving group. And if you look at that, that's basically just ADP as a leaving group. So electrons kick down, leaving group leaves. That gives you the product where the phosphate is attached to carbon 3 of glycerol. Oops. Got ahead of myself, but that's okay. Giving you glycerol 3-phosphate as your product plus ADP, which can go somewhere to the electron transport chain and be recycled to the ATP synthase in mitochondria and be recycled to ATP. All right, we're going to see this kind of thing with a kinase happen again and again and again. I'm not going to draw the mechanism for you in the future for this kind of step. I'm going to simply refer you back to this where you've seen it before. Okay, yeah?
So is it the kinase that's making sure that the oxygen on like the bottom of the glycerol is attacking the phosphorus, or is it just sterics?
Right, is it the kinase that's determining which of the hydroxyls attacks?
Yeah.
Yes. If you look at glycerol, look at its structure, it is not chiral. Carbon 2 is not a stereocenter. From 351, you would have learned that carbons 1 and 3 are enantiotopic, that those glycerol groups are enantiotopic, which means they're identical to each other except in a chiral environment. And what that means is the chiral enzyme, which is composed of chiral amino acids, can distinguish between the OH group on carbon 1 and the OH group on carbon 3. Once you put that phosphate group there, now you've got stereochemistry, and so the enzyme can determine that. It's not going to matter because even though we've made something that's chiral here, I haven't showed you stereochemical configuration because I didn't bother looking it up. It probably is known. But that actual stereocenter on carbon 2 is going away anyway. We're going to oxidize it.
Okay.
Yeah. Others? On the other hand, no? Okay. So that's the kinase step, and most of these phosphoester making steps where we attack the gamma phosphate group of ATP are going to involve this addition step, kind of like carbonyl chemistry or nucleophilic acyl substitution, and then elimination step. And as I said, some people are going to gloss over that and just show you it like it's an SN2 reaction, and you can be okay with that as well. But I've shown you what your text shows you. All right. Let's do now the oxidation step of glycerol 3-phosphate to dihydroxyacetone phosphate. And the enzyme that catalyzes this reaction, and you'll get good at, in biochemistry, you're going to get good at recognizing what an enzyme does based on what the name is. It actually gets pretty straightforward. Often the name of the enzyme substrate, or what molecule the enzyme is acting on, is in the enzyme name. This is called glycerol phosphate, because that's the starting material, dehydrogenase, or dehydrogenase, depending on how you want to pronounce it. Any enzyme that's named dehydrogenase is going to do redox chemistry. Another irritating thing about enzymes names is that enzymes, because enzymes don't affect thermodynamics of a reaction, they just speed the reaction up. Enzymes do not care which direction the reaction's going. So sometimes you just got to pick one direction and name the enzyme. So some enzymes are actually named for the backwards reaction. The kinase enzyme we just did is named for the forward reaction. Kinase is transfer of a phosphate group from ATP to some nucleophile. You're going to see some enzymes still named kinases that will do the backwards reaction, transfer a phosphate group from something to ATP. You're also going to see, in this case, glycerol phosphate dehydrogenase. You're going to see that glycerol 3-phosphate loses hydrogen. Just compare the molecular formula of those two molecules and you'll see that what's missing is hydrogen. We lose a hydrogen from the OH group on carbon 2 and we lose the proton that's on carbon 2. That's an oxidation reaction of glycerol 3-phosphate. But you'll see that some enzymes that do reduction reactions are still called dehydrogenases because they're named for the backwards reaction. So that can be a little bit tricky. The starting, you need a cofactor to help this process along, and the cofactor is NAD+. You've seen this before. We actually talked about NAD+ as an oxidizing agent. We've also talked about NADH as a reducing agent. I'm going to do something here which I hate, but which is involved in making sure that the charges are the same on both sides of the equation. All this means here, the comma and the proton, means that we need a base to remove a proton at some phase. Biochemists do this all the time. It irritates the snot out of me because protons don't just fly off into space, especially not in water. I guess they do in the gas phase, but this is all occurring in cells and in water. All that means is somebody had to remove a proton to make that happen. And the proton and the hydrogen that's on NADH, those constitute basically H2 that was removed from the molecule. So between these two portions, NADH and the proton, that's kind of like removing hydrogen gas from the molecule. So that's why it's called a dehydrogenase. Okay, so let's show you how this happens. After the reaction's done, this is called dihydroxyacetone phosphate. 3-carbon ketone is called acetone. You've got 2 OH groups, one on carbon 1, one on carbon 3, and the one on 3 is a phosphate group. Okay, so let's show you how this process happens. Drawing this mechanism is actually pretty easy. I'm going to erase these numbers just to give us a little bit more space. I'm going to use pink for the proton that gets removed by some active site group in the enzyme, and I'm going to use orange for the hydride that gets transferred to NAD+. You can look up the full structure for NAD+ somewhere. It's in your text. You can also just Wikipedia it. The business end of NAD plus is this nicotinamide end, which is what we'll draw with a positive formal charge on the nitrogen. And one of the things you notice about NAD plus is that benzene ring, or rather a pyridinium ring, is aromatic. Though it's got, this is a carbon-nitrogen bond here. I'll just put in parentheses, this is carbon 1 of ribose. It's the anomeric carbon of ribose. So that nitrogen, even though NAD plus is aromatic, it's sort of got that permanent positive charge in the ring. You can't get rid of it by removing a proton because, of course, the R group here is a carbon. Another thing that might be useful for you is to draw a resonance structure which would show you that the positive charge on the nitrogen is also on this carbon that is para to the nitrogen. So, and you can interconvert those two resonance structures simply with these two arrows, right? So that tells you that this NAD plus is electrophilic on that paracarbon. So what can happen is a base from the enzyme removes the proton from the OH group on carbon 2, electrons kick down, and blasphemy, H minus is going to leave as a leaving group. Now, that's a terrible thing to do if H minus were just going to float off on its own. H minus is not a good leaving group. However, the enzyme holds this NAD plus really, really close, actually touching that hydrogen atom. So it's not leaving and floating off on its own. This is a direct handoff of H minus from the substrate glycerol 3-phosphate directly to NAD plus. In other words, this positively charged carbon is just standing there waiting for the H minus to come off. So we'll show that as H minus is delivered to the carbon that's para to the nitrogen. We can do that from either of these two resonance structures. Either way, it's totally fine. Those are two equivalent ways of drawing the arrows. Your text will show you one. I don't care whether you do that or the other one. They're both equivalent. After the reaction, then we're left with not NAD plus, but now NADH, where that paracarbon is now Sp3 hybridized. And I guess, you know, here's the proton that got removed. Here's the conjugate acid of the base that removed the proton. It's not particularly important which of these two protons came from NADH. I'm just picking one of them. You can do labeling experiments with deuterium to demonstrate which proton it was that got, which of those hydrogens was the one that got transferred from glycerol 3-phosphate, but we're not going to worry about that. So here's the NADH, here's the H plus, and here's your dihydroxyacetone phosphate. And this is an oxidation reaction, or rather, the dihydroxyphosphate got oxidized and the NAD plus got reduced to NADH, okay? All right, we're gonna see a lot of examples of oxidation reactions like this. And I won't draw the mechanism for you in the future on those. I'll simply refer you back here because we'll push arrows in exactly the same way. The enzyme's going to be different. The base that removes the proton might be different, but this is basically how biology is doing these kind of reactions. Okay, questions about that? I guess I'll point out that you can sort of see why NAD plus is a good oxidizing agent, because it's aromatic, but it's permanently positively charged, so it's okay if it picks up a proton. But you can see why NADH is a good reducing agent, because if it loses H minus, it gets to be aromatic again. So there's this sort of balance. In its reduced form, it's at least neutral, but it's not aromatic. And in its oxidized form, it's aromatic, but not neutral. So there's a trade-off from going from one side to the other. And that's part of what makes NAD plus / NADH a good redox reagent. NADH can be used for a lot of things in biology for reduction reactions, but it can also be used in the electron transport chain to drive the proton gradient that leads to ATP synthesis. All right, so that takes us through what happens to glycerol. Now we need to do what happens to the fatty acid portion. And so what we'll talk about now is called fatty acid beta oxidation. And this is cool because it occurs in sort of iteratively, in sort of repeatable steps. And there's a chemical logic to it that makes sense. All right, so first we have to convert the fatty acid before we begin the iterative portion. We have to convert this long chain carboxylic acid into a fatty acid thioester. And actually you've seen this chemistry before as well. First, we're going to take ATP and make a fatty acyl AMP where the carboxylic acid attacks the alpha phosphorus. And again, the enzyme is going to direct that process of ATP. It's going to attack the alpha phosphate group of ATP to give you this intermediate in which the carboxylic acid has formed basically a mixed phosphoanhydride with ATP. And basically the idea is, as we did in the chapter on amino acid synthesis, whenever you want to do chemistry with a carboxylic acid, you first have to turn the negative, or carboxylate, you first have to turn the OH group or the negatively charged oxygen into a good leaving group. And that's sort of what we've done here. All of this is now a good leaving group. So how does this happen? I'm going to copy the structure of ATP that we saw from earlier. But you're going to see that the carboxylate attacks not the gamma phosphate group, but the alpha phosphate group. So we'll take lone pair electrons on that carboxylate. We're going to attack the alpha phosphate group. We're going to make a pentavalent intermediate as we've done before. We're going to do this via an addition step followed by an elimination step. Here is the new bond with phosphorus that we just made. Here is the alpha phosphorus, and it's going to be bonded to 5 things, except I'm getting stuff wrong. So This is the 5' hydroxyl of ribose, and this is the adenine base. And then over here is going to be the beta and the gamma phosphate groups, probably bound to a magnesium ion. Okay, that's what we get when the carboxylate oxygen attacks the phosphorus. This is the pyrophosphate leaving group. And at this stage, we'll show electrons on the oxygen kicking down and kicking off the pyrophosphate leaving group. And that's going to give us our product. PPI here is an abbreviation for the pyrophosphate leaving group. And you'll learn in biochemistry that this is a frequent leaving group, and it's nice because it can actually itself break down via a hydrolysis reaction into two individual molecules of phosphate. You'd get that if water attacked one of the two phosphoruses. And that can be an additional driving force for the reaction, though we won't consider that here. Okay? So if you're watching what we're doing, we basically have taken the oxygen from the carboxylic acid group and we have replaced it with something that's a good leaving group. And this is a very common way to do that in biology. This would be called a fatty acyl AMP, because you have adenosine monophosphate bonded to the acyl group of the carboxylic acid. Okay? And then let's just get to the point where we have the thioester, and then we can take a break. The next portion is to take the fatty acyl AMP as your starting material, where you basically have a good leaving group. And then you're going to use coenzyme A. I'm not going to draw coenzyme A for you. It's actually quite a long group with a thiol on the end of it. We could look it up. You should look it up once and see it in Wikipedia. You'll see it in your biochemistry textbook. You're going to see it and think, is all of that really necessary? And the answer is yes. The enzyme has to have something to hold on to get the thiol in the right place. But we're not going to draw it because we would be here for quite a while if we had to draw it each and every time. The mechanism here I'm also not going to draw for you because it is basically just nucleophilic acyl substitution. And if the thiol is the nucleophile, and I've shown you that the AMP group is the leaving group, you can basically figure this out. I'll get you started and then you can fill in the rest in your mind. A base removes the proton, the thiol attacks the carbonyl carbon, you get a tetrahedral intermediate, and so on. In the end, the sulfur from coenzyme A is bound to the carbonyl carbon of what was originally our fatty acid, and now we have this thioester. Okay. Questions about that? All right, let's take 5, and then we'll go through fatty acid beta oxidation and move on to glycolysis.